Let's understand the process of transcription in eukaryotes. As we know, the transcription process is a very simple process where the copy of the genetic information from DNA to RNA takes place. Now, just to note and just to recap what we have done in the transcription in prokaryotes, here the template strand of the DNA runs from 3' to 5', antisense strand would run from 5' to 3', and this would be the same as the strand for RNA polymerase. Now RNA polymerase would be required for initiation and elongation, but in the case of eukaryotes, enhancers are also required. In the case of eukaryotes, the nucleus is well refined and therefore the transcription process would occur in the nucleus. The structural genes here would be monocistronic, that means it encodes only one polypeptide at a time. So the transcription unit would have a promoter, terminator and a structural gene. This structural gene would be what? This structural gene would be monocistronic and this would be the RNA coding sequence. The end of the promoter would be the point where initiation would take place. The terminator would be the point where termination site would be located. So the first thing that we would involve in the case of eukaryote is the process of initiation. So again it would be held in three processes: initiation, elongation and termination. We'll understand these one by one. The first one is the initiation. In the case of eukaryotes, there are two sequences, -25 and -75, right? So -25 sequence is what is known as the TATA box and the -75 is known as the CATA box. So that's what you need to remember, the first thing. So the promoter here would be the TATA box and this is where you would have the start point. So the sequence would be TATAAA and that would be the site. So the promoter would have a TATA box and a CATA box. The next would be here in the promoter zone, the transcription factors would take place. So there are three polymerases. What are those polymerases? RNA polymerase 1, 2 and 3. So RNA polymerase 1, 2 and 3. And what do they bind to? So RNA polymerase 1 would transcribe rRNA. RNA polymerase 2 would transcribe mRNA and hnRNA, and RNA polymerase 3 would transcribe tRNA and 5S rRNA, right? So that's about the three RNA polymerase. So there are various factors that must bind to DNA before RNA polymerase 2 binds to it. So here would be the various factors that would be there. Then the additional factor after RNA polymerase 2 binds to it, which helps in the complete initiation process. Now again, the sigma factor here in the case of eukaryotes is not present and the primer is again not required. So what is important? Just before the initiation, RNA polymerase and the accessory proteins would bind to the DNA. So the RNA polymerase as well as the accessory proteins would bind to the DNA and then it would lead to the process of initiation. Now once the initiation point reaches, RNA binds to the promoter sequence. So RNA would bind to the promoter sequence and this would be the point where initiation of transcription would take place. Now as RNA binds to the promoter site, the polymerase would begin to synthesize the strands of the RNA complementary to one side of the DNA. So if this is one side of the DNA, the other side of the RNA would be synthesized based on the process of initiation and then the process of elongation would take place. So as you can see, here is the DNA strand. Unbinding of the DNA strand takes place and the new RNA strand would start to bind with the existing DNA which was untwisted in the specific region. And the region where it would bind is known as the DNA-RNA hybrid and most recently it is 16 to 18 BP, which is still bound to the DNA because it is still part of the DNA. It's not separated from the DNA. But this process is elongation. Why? Because the nucleotides are added to the 3' site and they are continuously attached one after the another. So another RNA polymerase would attach to the promoter to begin the synthesis, then again would attach to begin the process of synthesis before the first one gets finished over, and this leads to elongation. So lengthening of the RNA molecule would take place, and finally is the termination. So here there is nascent fall off or the process of termination. The RNA polymerase would be released and the sequence would be stopped. So termination is triggered when RNA polymerase actually encounters a DNA sequence, and this causes the polymerase to actually lose its affinity to bind with the DNA molecule. So here RNA is no more interested in binding itself to the DNA. RNA loses its affinity to bind to the DNA and separates off, and therefore there is release of RNA polymerase. Now at the point RNA polymerase is released, it disengages from the DNA and the RNA is not released, it is disintegrated from the DNA. There is release of the RNA, and this RNA molecule which is released for the purpose of translation and post-transcription processes would be taken into account. So the process of termination is also what is known as coupled transcription. Why it is known as coupled transcription? The idea is very simple, because the translation is not possible in eukaryotes. Translation was possible only and only in prokaryotes. So RNA passes from the nucleus to the cytoplasma for the purpose of translation. The nascent RNA or what is known as the hnRNA undergoes modification and it forms a functional mRNA. So it would form from the nascent RNA, it would become a functional messenger RNA. Now under the post-transcription modification of the nascent RNA, there are three steps. First is capping, tailing and splicing. So under the capping, what would happen? There is addition of MGPPP, which is methyl guanosine triphosphate, and this protects mRNA or messenger RNA from the release of nuclease. Now when it protects from the release of nucleases, it would provide a binding site for the ribosomes. The next is the residues would be added with the help of polyA polymerase, and it would protect the 3' site. Now also the last point is the slicing. So here as you can see, the introns would be removed from the main channel, and that would be by the process of splicing. Now again, if they are to be joined, they would be joined by the ligase. So this is again an important aspect that we need to understand in the process of post-transcription modification. So under post-transcription modification, three important elements: capping, tailing and mRNA splicing, of which splicing is the most important because it's point here where the introns are removed and they are joined by ligase in a definite order. So again, a very important step in the post-transcriptional process. In the case of eukaryotes, there would not be any process of translation. The transcription would take place in the nucleus. The structural gene in the center would be monocistronic because it actually is a single polypeptide which is present, and that's the basic difference between the process of transcription in the case of prokaryotes versus eukaryotes. However, the process of initiation, elongation and termination remain the same. In the case of eukaryotes, there would be the use of enhancers with the promoters, and three important polymerases here involved would be the RNA 1, 2 and RNA polymerase 1, 2 and 3. So RNA polymerase 1, 2 and 3 are highly important in the case of the eukaryotes, which are not seen in the case of prokaryotes. I hope this is clear. So this was a quick summary. For more details and detailed lectures, follow the link below. Thanks for joining in.